Okay, thank you very much. For those of you that are following, this is about the third year that we've done this type of a format where we've dedicated an entire day to mainly dealing with CWD and what we think is, we're trying to solve this problem. We've been chipping away at it for 20 or 30 years, and some days it feels like we're not making any progress. But I think one of the initial objectives of this was to bring up the newest and best information that we could to keep our producers informed, but also to develop a communication between all the different players that are involved in this. We have obviously ourselves, the farmers, but we also have wildlife, we also have government, and I think we came to the conclusion that pointing fingers at one another and trying to say, well, you're to blame, wasn't solving any problems. So having this forum every year has kind of brought everybody together, and I think it's been a very positive experience. Like I said, we've had what we think are the top people in the industry that are coming and giving us presentations. Again, we're always looking for new and better things, so as I said, if you've got speakers that you want to suggest that belong here, we're always open to hear that. One of the up-and-coming topics that has evolved out of this is the whole idea of different strains of CWD, and having heard Glen Telling give us a little presentation on it about a year ago, we decided that he was someone that was worthwhile for the rest of everybody in the association to hear. So I'll let him take it away from there and give us a warm welcome, and we'll go from there. Thank you.
All right, good morning everyone. It's a great pleasure to be here back in Saskatoon. We're going to talk a bit of science first thing this morning. So, and unfortunately it's a very challenging area of science, so I'm going to try and keep my comments as straightforward as I possibly can. But unfortunately you are involved in one of the most interesting and dynamic and controversial areas of biology, and that is the prion world. So my comments are going to be in the context of prion biology, and what I'm going to talk about is specifically chronic wasting disease, its host range and strain properties, and tell you about some of the methodologies that we've used to kind of investigate the unusual biology of these infectious agents. We're going to be talking about transgenic mouse models, we're going to be talking about strains, and hopefully this will give you a bit of background to kind of understand where we are in terms of the basic research in the field. So chronic wasting disease, as you all probably know, belongs to a group of diseases, related diseases called prion diseases, otherwise known as transmissible spongiform encephalopathy. The prototypic prion disease is scrapie. It's been known as a disease of sheep and goats for some 200 to 300 years. Mad cow disease, BSE, is also no stranger to Canada. Disease of cattle and also exotic ungulates that had major economic, political, and public health ramifications. But chronic wasting disease is really a disease that is our major focus today. It's a disease that is expanding in range, and it's a disease that's not going away. It's unique in that it's the only known prion disease of wild as well as captive animals, and it's extremely contagious and therefore has been very difficult to control. The other thing that you need to know is that this is not a static picture. This is an expanding universe, and we, as the more we look, the more we understand that there are new strains and new forms of prion disease being discovered all the time. So most recently, we just discovered, or actually a group in Italy discovered a new disease in camels called camel prion disease. So this is just to exemplify that we cannot be too complacent about this. So BSE, just to give you a bit of background, was a disease of cattle, and this is the BSE epidemic in Great Britain here. First identified in the mid-1980s, when it became clear that this was caused by feeding contaminated meat and bone meal to cattle. there was a feed ban imposed, but you can see here it took about 5 or 6 years for this ban to be effective, and so the disease peaked at this stage, and now it's, and this is obviously out of date, but now this is an extremely rare disease. But of significance here, it became clear in the mid-1990s and subsequently that this was a zoonotic disease. In other words, it could spread and did spread to human beings in the form of a new type of Creutzfeldt-Jakob disease called variant Creutzfeldt-Jakob disease. Now, you can see here that this is really exemplifying the success of science, and also the fact that we're able to control this disease not only in cattle but also in humans really testifies to the power of the prion hypothesis. The reason that this disease went away and that this disease was contained was because we understood this disease in the context of it being a prion disease. So all the diagnostic capabilities, all the therapeutic approaches, all the understanding of the biology of the prion diseases were really successful in controlling this disease, but in the context of it being understanding that this is a prion disease. And I think the same is going to be true of chronic wasting disease. So just a bit of humorous background, as we probably all know, for those of you that have been following this debacle, this descent into hell, which is called Brexit. Today is supposed to be Brexit Day, but of course it wasn't. And there's an interesting correlation here that we found on the web between the voting pattern that occurred for Brexit leave, which is shown, I think, in blue here, and the incidence of BSE. And so I think this is a correlation that probably needs to be examined a bit more. Obviously, this is a joke, but anyway. Okay, so just to get back to some of the more unusual features of this disease, that to put it into sharp focus, it's been known for many, many years that the biological properties of these infectious agents were very unusual. So, for example, this researcher, Tikva Alper, who was a radiation biologist, showed for the first time that prions were basically resistant completely to, in this case, UV dosing, and what was found was that UV treatment that would otherwise inactivate other infectious agents had no effects on the scrape agent or prions in general. And this led to the idea that these agents were devoid of nucleic acids, and so put them really into a unique class because UV treatment destroys nucleic acids. And so I'm summarizing obviously decades and decades of research and just exemplifying one unusual component of these diseases, but this led to a real problem in the field. So how can you replicate infectivity in the absence of nucleic acids? Because all infectious agents that have been known to date really required nucleic acids, DNA or RNA, to actually make a protein and encode strain information. And so somewhat later in the 1980s, this chap, Stan Prusner, who I was fortunate enough to do my training with, discovered these infectious proteins, which he used the term prion to describe them. Instead of finding viruses in these purified preparations of infectivity, he found instead this protein which had properties that were unusual, and they tended to aggregate, and I'll talk a little bit about that today. So his hypothesis, for which he received the Nobel Prize in the late 90s, was that prions, these proteinaceous infectious agents, are devoid of DNA or RNA, any information on nucleic acid, and therefore differ from all known infectious agents. And this is true of chronic wasting disease, human prion diseases, BSE, scraping. And the idea here is that prions are composed largely, if not entirely, of this abnormal protein which is called PRP scrapie, and this is an abnormal infective isoform of a normal protein that animals and humans express, and that normal protein is called PRPC. Everybody in this room expresses this protein normally, but somehow during disease, something bad happens to transfer this, to change this protein into an infectious form. And that's really the crux of the issue here, and what makes one of the major things that makes these diseases really unusual, and why several Nobel Prizes have been awarded so far. Fortunately, the biochemical properties of this form, the infectious form, and the normal form are subtly different. So for example, the infectious form is partly resistant to proteinase treatment. And this is useful for us because it gives us an opportunity to be able to identify the protein, and it's really the basis for most diagnostic procedures that involve detecting the prion protein. So just in cartoon format, the idea here is that the shape or the conformation of the protein that we all express here during disease changes from the normal conformation, the normal shape, into an abnormal conformation, which is infectious, and induces the normal form to change into the abnormal form. So, and the idea here is that these aggregates form during disease, these aggregates of PRP scrapie, which I'm showing here in squares, and the notion is that the normal form gets recruited into these growing aggregates and is converted, the circles become squares, in other words, and that somehow during the propagation of disease that there must be also breakage of these growing aggregates in order for replication to occur. So one of the major problems that needs to be addressed here is that, like viral diseases, like bacterial diseases, like all known infectious agents, really, prion diseases can also exist as different strains. So like influenza, we know that there are different strains of influenza every year. There are different strains of herpes viruses. And the idea behind strain variability in viruses is that there are subtly different changes in the nucleic acid component that encode strain differences. So I've just told you that there are no nucleic acids in the context of prions. So how can we have strain differences occurring? Just by way of introduction, in the prion world, we define strains in terms of their differences in incubation time. So these three strains have different times to onset of disease in an experimental setting, and also the different neuropathologies that they produce in the brains of infected animals. So how does strain information get stably passed from one generation to the next in the absence of nucleic acid? For those of you that aren't scientists in the audience, it's just, I think, sufficient for me to say that the shapes of these molecules here, as shown by this western blot, are subtly different, and this is reflected by slightly different migration patterns on a western blot. You can see this is slower than this one, and this gives us an idea that the conformation of these two different strains are different. And so this leads to the idea that prion strain information is somehow enciphered or encoded within the conformation of PRP scrapie. So the shape of the molecule is subtly different from one strain to the next. So, what I told you so far, prions are unusual pathogens. They lack nucleic acid. Their replication involves the induction of the pathogenic conformation upon PRPC by the pathogenic conformation PRP scrapie. The SE prions already have caused disease in young adults and teenagers in the UK and Europe in the form of variant CJD. The risk to humans from CWD prions in the US and also now globally remains unclear. And I've also told you that prion strain information appears to be encoded in different conformations of PRP scrapie. So another concept that I think is important to introduce here is the so-called species barrier. So although infection is transmitted readily to animals of the same species, in other words, in the case of chronic wasting disease from one deer to another deer or from one elk to another elk, interspecies transmission is generally more difficult. So the question is, for example, could CWD prions in elk or deer transfer into cattle or sheep? And this is obviously the concept surrounding the species barrier. Experimentally, we can look at this in the following way. We can try to inoculate, for example, in this experiment, human prions into normal wild-type mice. And when we do this is generally a very inefficient process. Most of the animals that are infected at day 0 fail to get disease even after many hundreds of days. But sometimes, at a very low rate, you can get disease being induced. So the next part of this experiment is that when you take the materials from the diseased animals that actually contracted disease after several hundred days inefficiently, then retransmit that material back into the same genetic background, in other words, wild-type mice, this now becomes an efficient process. In other words, there's been some kind of adaptation process happening here. So these human prions have adapted across the species barrier and are now making mouse prions which can cause disease very efficiently on second passage. So this led us to a new breakthrough, which I was involved in many, many years ago now, more years than I care to remember. But we decided that, and this is based on the work of Mike Scott, who was also working with Stan Prusner, we decided to express, by genetic engineering, the prion protein for human, PRP, in mice, so-called transgenic mice. And the idea was that by doing this, we could eliminate this species barrier. And in fact, when we now express, instead of wild-type mouse prion protein, which leads to a very inefficient process, if we now express the human protein and we inoculate with the same prions, this goes from being an inefficient process to being an efficient process. The reason is because we're expressing the human prion protein, which will now convert in response to infection with human prions. So we can, in other words, very simply speaking, by doing these kinds of genetic manipulations, eliminate the species barrier and now begin to be able to study prions from different species, in this case, human prions. Okay, so I'm going to switch gears now and talk about chronic wasting disease. This is a disease of cervids in North America. Clinical signs include gradual loss of body condition, behavioral alterations, generalized incoordination. The clinical course is slowly progressive, much the same as other prion diseases. I don't need to go into too much detail about this, you're all very familiar with this. It's the only known prion disease of wild animals and it's highly contagious. Prions replicate to high titers not only in the central nervous system but also in the lymphoreticulae system of infected animals. We can detect prions in saliva, blood, urine, feces, and velvet, and also in skeletal muscle, as I'll show you in a minute. And experimental transmissions are extremely challenging in a natural host because they take a long time, and obviously experimental studies in deer and elk are very difficult. I also probably don't need to spend too much time on this. This is a disease of expanding geographic range. It was first identified in the so-called endemic region in Colorado and Wyoming. First by the late Beth Williams in Fort Collins in captive animals, and it was then detected in wild animals in Rocky Mountain National Park. And this is the kind of timeline here, and you can see here that it was imported into Canada inadvertently. It's spread now to not only western states, but also in the early 2000s to Wisconsin and eastern states. And you can see here now this is the state of play. Currently about 26 states are known to have chronic wasting disease either in wild or captive animals. And my recent counting indicates there are 3 Canadian provinces that also have detected CWD. This has become a global epidemic. CWD was inadvertently imported into the Republic of Korea in the early 2000s. And in 2016 it was first diagnosed in moose in Norway and also subsequently in reindeer and red deer. In 2018 CWD was diagnosed in moose in Finland, and 4 days ago a report was released that CWD had been detected in, not surprisingly, moose in Sweden. So this is truly a global epidemic at this stage. So the host range of CWD is also expanding. So in addition to being diagnosed, first of all, in mule deer and later Rocky Mountain elk, it was actually first identified in retrospective studies in black-tailed deer in a zoo in Ontario. White-tailed deer preferred hosts in eastern states. This is the most frequently encountered species. Moose were first diagnosed in the mid-2000s in Colorado, and red deer was the most recent species to be detected in Quebec. In addition to its importation in mule deer and elk into the Republic of Korea, these endemic species, Sika deer and red deer, also became infected. And also I've mentioned that moose, reindeer and red deer have also been diagnosed with CWD in Finland, Sweden and Norway. So when I got into this field, after doing work on human prion diseases that I described earlier with Stan Prusner, this was the state of play pre-2000. The only recognized disease of wild animals. It's highly contagious, its geographic range is expanding, its host range is expanding. Its origins are completely unclear. The means of transmission was, at that stage, unknown, and its host range also was unclear. The prevalence of strains in the wild was completely unknown, and the risk to humans from exposure to CWD-infected meat and other tissues was also unknown. So based on these unknown questions, we decided to use the same approach that I described earlier to generate transgenic mouse models. In that case, they were for human prions, but to use the same approach to generate transgenic mouse models so that we could study CWD in transgenic mice in the laboratory. And so this is the study that we published in the early 2000s, I think, many years ago, 2004. My graduate student, Sean Browning, was involved in this work. We made these mice expressing deer prion protein, which we call 1536. They overexpress the deer prion protein, they fail to express the mouse protein. And just to summarize, when we infect these mice, either by direct infection into the brain or to the brain, we can induce disease in these mouse models. So for the first time, we could really study chronic wasting disease in a laboratory setting without having to resort to the natural host. So one of the first things that we did was to ask, because obviously these animals are hunted and their materials are consumed, whether or not we could detect prions not only in the brain but also in skeletal muscles of deer infected with chronic wasting disease. And this is to show you that when we inoculate these animals with the brains from animals with chronic wasting disease, the incubation time is around about 200 to 300 days of age. When we take skeletal muscle from those same animals and we inoculate them in the same way, disease is also produced, but with a slightly longer incubation time. What this tells you is that in addition to infectivity being in the brain, we can also detect in diseased animals infectivity in skeletal muscle. And so this told you for the first time that persons consuming this material would be at risk of being exposed to CWD. $64,000 question is whether or not these prions would cross the species barrier and cause a new disease in human beings in the same way that it did with mad-cow disease, or BSE. And what I can say is right now there's no evidence for that, but obviously this is an area of investigation that is under intense scrutiny right now. So I want to talk a little bit about some other methodologies that have been produced over the years for studying prions. One of them is called PMCA, protein misfolding cyclic amplification. This is, for any of you that work in this field, it's a bit like PCR. It's a way, in the PCR we can amplify nucleic acids and start with very small amounts of nucleic acid and amplify it using enzymes. PMCA is a way of doing the same thing with the prion protein, obviously without the involvement of nucleic acid, but we use sonication to, without going into details, to amplify vanishingly small amounts of signal, and we can amplify and detect it very readily. One of the first things that we did was to amplify CWD, inoculate the same transgenic mice, and show that the transmission properties were not changed upon amplification. And this is showing you this here. This is chronic wasting disease, which will be familiar to many of you. This is the neuropathology that we see when we inoculate these animals with CWD. This is the brain, this is the hippocampal region. You can see these massive amyloid plaques that are induced in response to infection. We see the same thing happening when we amplify by PMCA, and we inoculate that material into mice here. And this is another way of showing that there's no difference between the normal CWD from infected animals infected into these transgenic mice and PMCA amplified material. Okay, so the next thing we wanted to ask was, could we detect different strains in the wild, in different species and in different geographic locations? So with the help of our collaborators in Canada and Wisconsin and in the endemic region, we took a large number of different isolates and we inoculated our transgenic mice, and we looked to see if we could see any differences in the disease profile that would be indicative of different strains. And what I can tell you is that, and this is published now almost 10 years ago, that we detected two different strains which we imaginally refer to as CWD1 and 2. CWD1 has a very short, relatively short incubation time. It also produces this sort of bilaterally symmetrical distribution of the prion protein, which is being shown in these pictures here. This is again the pathology that we see when we infect these mice. And we see a different pattern in this strain, which we refer to as CWD2. The incubation time is much longer. The distribution is asymmetrical across coronal sections of the brain. And you can see here we get these massive aggregates of PRP scrapie. So in this case, we identified two distinct strains that had the properties of incubation time and pathology that I indicated were the way that we define strains that were very different. Okay, so you're going to hear quite a bit today about protective polymorphisms in the gene for the prion protein, and using the kinds of approaches that I've already talked about, transgenic mouse modeling, we're able to study the effects of these polymorphic variations by expressing not just the wild type gene, but also the polymorphic gene in transgenic mice. And what I can tell you is that these protective polymorphisms have a significant effect on the development of the disease. So this is the wild type, transgenic mice expressing the wild type gene. This is transgenic mice expressing equivalent levels of a protective polymorphism at 95. And you can see here that these animals fail to develop disease within the same time frame, in fact, for many hundreds of days afterwards. So the genetics of the prion protein gene can be very useful in identifying relatively resistant animals in the population. And these experimental approaches really confirm that that's the case. Okay, so I want to talk now about some currently unpublished work, but we're quite excited about this. So I told you so far that we generated transgenic mice expressing actually not only deer prion protein, but also elk PRP. This is important because deer and elk PRP, the genes for these prion proteins are exactly the same, except at one residue at 226 here. So moose, caribou, and deer all have glutamine, shown in purple here at residue 226, whereas elk have glutamate at this position. We don't need to remember that or be concerned too much about the details here. But this genetic variability is interesting, and we really wanted to know whether it had an effect on the pathogenesis of CWD. In other words, whether disease would be different in deer compared to elk. So in order to do this most precisely, we made different mouse models, and these are so-called gene-targeted mouse models. And I won't go into the details of this, but it's a much more precise way of studying the genetics and the expression of the prion protein in mice. Just to suffice to say, these animals express exactly the same levels of deer or elk PRP, and they only differ except at one residue at 226. So these mice expressing Q are different from mice expressing E only at residue 226, and they express exactly the same levels of the prion protein gene. Now this is also convenient because we can make heterozygous, in other words, one copy of each Q or deer or elk PRP in the same animal. And we can also manipulate the levels of expression in ways that I don't need to go into in too much depth here. This is not a take-home slide by any means, we inoculated these mice with a large number of different deer and elk samples and moose and so on. And this is just to summarize all of our results, and this is under review right now. What we found here was that these mice expressing Q at 226, in other words deer PRP, the kinetics of disease onset was slower than mice expressing the elk prion protein. Now remember, these mice express exactly the same levels of the prion protein, and so the difference in kinetics, which is significant, very significant, is due only to this residue at 226. So this tells us now, for the first time, that the pathogenesis in deer and elk is expected to be very different. So we also made heterozygous counterparts of the E and the Q, and we found that the heterozygous were intermediate in incubation time. Okay, so we have now both transgenic and gene-targeted mice here, and this is the incubation time here on the y-axis. These are the different animal models. And this is just to show you that the level of expression, the incubation time, the more rapid incubation time is linked to animals which have the highest levels of expression. This is not too important to understand, but it's certainly significant. Okay, so I told you that one way that we can study strain variability in prion diseases is by looking at the conformation of the prion protein. So we were very interested to look at whether or not these animals expressing either elk or deer PRP had produced prions with different conformational properties. And so this curve here is just a way of us looking at this. It's a so-called guanidine denaturation curve. The details of this are not important. But when we compared these curves in the deer gene-targeted mice with the curves in the elk gene-targeted mice, we saw a significant difference. So the conformational properties of the prions in these two different animals are radically different. And also, when we looked at the pathology in the brain, or the distribution of PRP scraping in the brains of these animals expressing either the elk protein or the deer protein, we saw very distinct differences. So this is the elk protein infected, elk gene-targeted mice infected with CWD. You can see a very diffuse distribution of prions, which is different depending on whether these animals are infected with deer or elk. And when we looked in the animals expressing the deer prion protein, you can see here that the distribution is very different. So what this tells us, we think, without going into details of these experiments here, and I'm running short of time, I'll just briefly summarize this, because I don't want to burden you. Well, what I will say is this. These animals, these gene-targeted mice, they showed us that for the first time we could detect the prions not only in the central nervous system, but also in the lymphoreticular system of these infected animals. So this reflects what we see in the natural host. This wasn't the case when we made transgenic mice. So there's something about these gene-targeted mice which is much more precise. It gives us a much more precise means of studying CWD and its pathogenesis without having to resort to studying disease in the natural host. In other words, it recapitulates exactly the disease that we see in either deer or elk, and those differences are real. The other thing that we were able to show here, if I have the slide, yeah. We all know that chronic wasting disease is a highly contagious disease, and so because these mice that we just made, that I just told you about, these gene-targeted mice, seem to recapitulate even the peripheral aspects of disease, we asked whether or not we could study contagious transmission of chronic wasting disease using this mouse model. So what we did was we inoculated a group of mice, and this is the elk gene-targeted mice, and these mice, as I've shown you before, developed disease at around about 250 days. And then after we inoculated these mice at early stages, we simply put those infected mice into new cages of the same mice that were uninoculated. In other words, they were just co-housed with infected mice. And we simply asked whether the disease in these infected mice could be transmitted contagiously to the animals that were co-housed with those same infected inoculated mice. And the answer is yes. We could induce disease by contagious transmission by simply co-housing these animals in the same cages with each other. So now we have a really useful model where we can study the contagious transmission of disease in a lab setting. All right, so there's a lot of information. I hope I haven't overburdened you with this, but I'll summarize what I told you already. Incubation times in these mouse models that I've described here, gene-targeted mice and transgenic mice, are related to levels of cervid PRPC expression, either deer or elk. I've skipped over this, but this is important. Residue 226, which differs between deer and elk, controls the kinetics of CWD onset, and also the pathogenesis, the deposition of PRP scrapie or prions in the brains of these animals. Also, the conformational properties of CWD prions in these two animals are different. So the way we interpret this is that residue 226, the residue that differs between deer and elk, is a major controlling element. It dictates the selection and propagation of distinct CWD strains. What I think this means is that it gives different species, deer or elk, the opportunity to propagate different CWD strains in the wild. Okay? So this is new information that we didn't know before. Peripheral pathogenesis in these gene-targeted mice is efficient. This is not the case in the transgenic models that I told you about. In these gene-targeted mice, prions replicate not only in the brain, but also in the lympho-reticular system, and this is a major feature of chronic wasting disease. And CWD transmission by co-housing infected with uninfected mice is efficient. So now, for the first time, we have a tractable model to study contagious transmission and also to devise therapies to prevent this from happening. All right, let me talk for a few minutes about CWD in 5 minutes, right? In Norway. And this is a collaboration with Sylvie Benestad, my collaborator in Norway. I told you a lot about North American CWD. We know quite a bit about that. And so when CWD was first diagnosed in Norway and subsequently Finland and now obviously Sweden, the question is the CWD that's in Norway, in Scandinavia, the same or different from CWD that we all know and recognize in North America? And the answer is it's very different. The strain properties of Scandinavian CWD are completely different from North American CWD. How do we know this? Well, I told you we have animal models where we can study the transmission properties of CWD. We can identify characteristics that are related to differences in strains. And when we compare the transmission properties of North American CWD, without going into too much detail here, you can see here that the transmission properties of North American CWD are very different from Norwegian CWD. Most strikingly, Norwegian CWD in moose fails to transmit into transgenic mice expressing E at 226. It only transmits in animals that express Q at 226. This is the opposite of what we see in North American CWD. We've done the same thing with reindeer prions. You can see here, this is deer, representative deer and elk CWD from North America. Transmits both into the deer transgenic mice and the elk transgenic mice. But the Norwegian reindeer transmits relatively efficiently into deer transgenic mice, but relatively inefficiently into the elk transgenic mice. There are other indications that the strain properties of the prions in Norway are different from North American. One is that I told you about our ability to recognize differences by Western blot. This is called a Western blot here. You can see here that comparing Norwegian moose to North American moose, that the migration of North American moose is very different from the migration of Norwegian moose. And also, when we look in the brains of infected animals, that the distribution of North American moose, you can see here, is highly punctate distribution of PRP scraping in the brains of these infected animals. It's very different from this much more diffuse deposition that we see in Norwegian moose, these two different examples here. And the same was true when we looked at, I think that's the same. I won't go into any more detail about this, except to say, and this is not published yet, but it will be soon, that to reiterate, the strain properties of Norwegian moose and reindeer are very different from the strain properties of North American deer and elk CWD and moose CWD. So the question is, where did this disease come from? If not North American CWD. The other question is, we all know that, as I've told you, that these prions can have zoonotic properties. They can transmit not just within species, but in the case of BSE, it's very clear that zoonotic transmission has occurred. there's no evidence so far, or at least there's conflicting evidence that the zoonotic properties of North American CWD, that CWD might be zoonotic, and these are studies that are ongoing. This changes everything. The question now becomes whether or not the tendency for Norwegian or Scandinavian prions to infect humans, is it different or the same as the tendency of North American CWD prions to do the same? So I think I will stop there and just thank my collaborators in Norway, in particular Sylvie Benestad, the people that actually did the work in my lab, in particular Jiffin Bien, Julie Moreno, Jeff Christensen, Seyon Kim, an ex-graduate student of mine, Vadim Kachuk, and Eri Saijo, who's now working at the Rocky Mountain Labs for NIH, Sarah Kane, an undergraduate, Joseph Gallegos, Heyun Kang did much of the work I didn't have time to talk about today. This was funded by the National Institutes of Health, and thank you for your attention. I hope I didn't go into too much depth and lose everybody, but thanks a lot.
Okay, we've got time for a couple of questions. Ian.
Could you generalize?
Are there field mics for these
Yeah,
guys?
right behind you. Behind you.
Behind me? Ah.
Thank you. Can you generalize? Can you comment in a general way? Is that working?
That's where we go.
Yep.
Good. Okay. In a general way, and I realize we haven't got probably time to really discuss this, but incubation periods for the various strains.
Yeah. So when we look at, I told you we did a study using our transgenic mice, and also more recently our gene-targeted mice, where we looked in the first case at over 30 different isolates from Colorado, Wisconsin, Wyoming, Alberta, I don't think Saskatchewan. We didn't, and we did not only initial transmissions, but also after stabilization, we serially transmitted that material. We weren't able to see any isolate-specific indications that different isolates had different strain properties. What we were able to see were these type 1 and type 2, but they seem to coexist in the same inoculum. Now, when we go back and we look at our gene-targeted mice, that coexistence doesn't happen. In other words, we don't get strain 1 and strain 2 coming out within the same transmission experiment. The question is why? So we think now, because those transgenic mice overexpress the prion protein, that could have led to artifactual emergence of strains during the experimental transmission. It seems as though the gene-targeted mice that express normal physiologic levels of PRP give a much more accurate representation of the strain, and we don't see that variability in incubation time. But to answer your question, when we've looked at various different North American isolates, we're not able to see any specific indication of different isolates of different strains, except to say when we compare in bulk elk prions compared to deer prions in those gene-targeted mice, the strain properties in those gene-targeted mice do appear to be different. So what that says is that that residue that differs between elk and deer is most likely selecting for the propagation of different strains in these different species. That's important because if CWD ever was to transmit to other species, in particular humans, understanding strain differences are crucial, because the strain properties of the prion really influence the ability of the prion to transit species barriers. So if the strain properties of CWD in deer are different from the strain properties in elk, we have to assess the zoonotic potential of those prions separately. I hope that's clear. Yeah.
If
If, hello?
Yeah.
There we go. If we can tell that there's prions in feces and blood and urine and antler velvet,
Yeah.
why are we having such difficulty with the live test?
Well, I don't know, Nick, are you gonna be able to talk about this?
I'm trying to clarify some of the stage that we're at.
Yeah, I'm not sure what difficulty. This is not my bailiwick, right? I don't do a lot of work in terms of understanding live testing. I'm not a veterinary scientist. I'm a basic researcher. We're trying to understand the nuts and bolts of how these agents replicate. So I'm not the best person. I'm not the most qualified person to talk about testing and so on and so forth. What I will say is that a lot of work has been done on the back of mad cow disease in particular to devise highly sensitive methodologies for detection of these infectious agents. So in other words, the PMCA test that I talked about, and also another test called RT-Quick. This was developed by Byron Coey at the Rocky Mountain Labs at NIH. The analogy that he uses is that you can take one molecule of the infectious agent, put it in the swimming pool, and still be able to detect whether or not you had an infectious agent in there. So these are highly sensitive tests. Now, clearly they need to be adapted for CWD, but I think this is the best opportunity that we have for moving forward in terms of detection. And that's really all I'm going to say, because I'm not an expert in field biology. So.
I'll just say, I think the limitations right now are primarily governmental agencies. I mean, I think some of these tests have had a lot of field work done on them. I mean, years and years. Most of you know I came up here. I'm sorry if you can't hear me, it's on, but most of you know I came up here a few years ago. We ran rectal biopsies on elk, and I think it's just a matter of getting the agencies to accept that and accept these new amplification technologies. It's kind of like prying it out of their hands.
Thanks.
One more question at the back.
Have you looked at any mouse model where you've got a mutation in both 95 and 226? Where you have an H amplification?
No, we haven't done that. H95 you saw it. It's pretty dead in the water. But we haven't combined. We did that in the context of Q at 226, not E at 226. So we haven't looked at. And the reason we did that was because that polymorphic variability occurs in the deer context of the deer prion protein, not in the elk, and that's why we didn't engineer it into the elk because it's not a naturally occurring difference. But yeah, we haven't looked at that yet.
Is that something in the future we could do?
We could look at that, yeah. It might be worth looking at.
I think in the white-tailed deer, it's fairly easy for us to breed that.
Yeah.
So if it's something that we can take a look at, kind of steer us a little bit in our breeding programs.
Oh, you're saying about the lysine at 226 and also the polymorphic variability at 95. Yeah, that's interesting. Yeah.
We'd love to see that data.
Yeah, good idea. Thank you.
Okay, thank you very much. And just a reminder, for everyone who can stay, I'm not sure who all is going to be available. There is a panel discussion this afternoon where if you've missed any questions, you can give them another shot. Leonard, I'm not sure, are you staying? Are you available?
My flight's at 3, it depends on what's
Okay,
on.
all right.